Okay, so in this step, we are going to use the glucomics to actually classify which cell lines belongs to what cell type. So in the previous exercise, we already basically clean up the data and then harmonize the CCLE and also the ICGC database. I mean the data set. Now, even that, if you look at the ICGC, there are still relatively quite a lot of genes here that we are looking at. It's about 13,000. So now, if you want, it will be while you can insert the whole data set into the glucomic to do the analysis, but then it will take a longer time to simulate because the number of data is quite high there. So what I'm going to do is now is basically to introduce another process is, okay, instead of putting in the whole data set, I'm going to input only the data set that are significant, okay, from your previous exercise when you do your C-score comparison. Okay, so basically, if if you remember when you do the C-score comparison, so eventually you have a list of genes that are considered up or down regulated. Okay, so these are the one. Now, so what I'm going to do now is basically try to match, okay, the data set here, the entry with the significant gene. So again, I'm going to, okay, so now since both are already harmonized, what I can do is I can actually merge them together. I want to, okay, copy and paste and then merge them under the same. So you are comparing C-score versus C-score, so now it is also become comparable because two data set was actually originally using different platform. Okay, so I will mark this as CCLE here. Okay, so what I'm going to do now is I'm going to mash this again, count if they appear in our gene list. So you have all the four subtypes, these are all the significant genes. Okay, if they match, if they appear in this list, then it will return as 1. Okay, I'm going to copy this down. So you have AICF1, which actually appear in the down-regulated genes in the squamous, and then you also have AAAS. So these are the ones that are actually not significant. Okay. Okay. So I'm going to I'm going to remove those that are not significant. So this one is fine. Yeah. Do you know why I say this one is fine with the number 2 here? Because what it means 2 here, that means these genes appear twice in your significant up or down regulated genes. So why? Because they could appear significant in 2 different subtypes. For example, in this case here, they are upregulated in the squamous as well as where's as well as oh, as well as downregulated in the pancreatic progeny. Okay, so that's why I will leave that in here. I don't need this column anymore. I can delete it. So now that will down to about 900 over genes that are significantly regulated in all the different subtypes. Okay, so now, okay, so now I'm going to process this and into the input file for the glucoamic. Okay, now glucoamic follow a certain input format basically. Uh, Let me save this one first. This would be the ICGC glucose. Okay, so now I'm going to insert the ICGC dataset first to build the model. So normally you have to create one column, extra column here, and also an extra row here. So this will mark. So this will refer to these two uh, rows, and then this will be the genes, right? Then when you save it, make sure that you are saving it as the CSV format. Okay, so now you can save that. Okay, now then you can go to glucoamic interface here. Okay, you can just open with Wizard. So if you are not familiar with it, you can always open with Wizard. They will ask you step by step to indicate what value is for what. Go to here. Where is the okay ICGC? So this is the CMV format. Okay, go to Wizard. So they will tell you you can read up about it. So basically, so it tell you okay what is the separation is by comma. So matrix orientation is by variables here and then sample across. Then they will tell you to mark the data matrix. So this is this is the beginning of the data matrix and then the end of the data matrix. So now you have this whole area which is your data matrix, and then you have the row variables which is here, and then you have the column variables or samples which is here. So now once it is done, then it will give you a PCA plot like this with all the 4 different subtypes based on what you have marked there. So okay, the PCA is a good thing to help you to see how separated they are. So if they are well separated, then you can see 4 different distinct group there. Okay, so in this case here, we are not too bothered about it. Now, the next thing what I'm going to show you is basically how to build the classifier to actually identify the subtype. Okay, what the classifier means is that basically based on your data set, we are asking the software, okay, can you tell me which variables can help me to predict correctly whether a particular tumor is belong to which subtype based on the pattern. So to build that, so you just go to, okay, build classifier. So you have 3 different methods, KNN, SVM, and also the random trees. You can read more about it. So I'm going to, so in the project, so basically I use 3 different models. So again, the principle is that if 3 models predicting the same thing, then the chances of it to be correct is much higher. So now, but bear in mind is that, well, when you use these 3 methods, okay, sometimes you will get a variable result. For example, if I do it now, okay, it will be somewhat different compared to what I did it previously, or could be somewhat different compared to what you would do it later on, because each of this algorithm there is a function of the randomness there. Okay, so if you want, you can do a permutation. That means you ask the computer to run 1,000 times of simulation. Okay, so and if 1,000 times it also come out to predict the same thing, then then it will probably correct. Sometimes you will come to sometimes you will come to the case is that, well, at one of the analysis, even though with the same input, okay, with one of the analysis, this cell lines is classified as okay squamous. In if you repeat the the same analysis that it come out as progenitor, then when you repeat it again, it come out in squamous and so forth. It could happen. Okay, those are what we call the classification is not stable. Okay, it could. In that case, then we will probably cannot really make a conclusion where it is actually belongs to. So again, these are prediction. Okay, so anyway, so I will show you how to do the KNN first. So we'll do KNN. Ranking will be using multi-group comparison and validation. You can choose. So in this case here, we don't have a validation dataset. So we will use the same dataset as input, okay, for validation. Okay, it will be it will be predict based on the class, okay, the subtype, and then you can just build. Okay, so okay, I can then save it here. Okay, so now this is how it will tell you. So you are using all the 96 samples that we have. No, okay. So the accuracy, so in terms of predicting class 1, so you have 100% accuracy. That means whatever it is predicted to be class 1, you probably have 100% accuracy of it. So if anything that's predicted to be class 2, you have 96% accuracy and so forth. So the mean prediction is about 93.5% accuracy, so which is considered quite good. Anything more than 0.9 is considered quite a good model. So now, and then here, these are the genes that, these are the panel of genes that you can use to actually do the prediction of the subtype. Okay, so now, yeah, I think that's good. So I will close this for the time being. Now I will go to the CCLE dataset that we worked on just now. So I'm going to create another. So I will also format exactly for the input to the what you call it, glucomics. Okay. In fact, if you want to do this, or another way you can do it, why don't I just do the combined dataset here? I can use the combined dataset. Okay, so this is a combined dataset with the ICGC cells, I mean the ICGC and also the CCLE data together. So this is the gene. Okay. Oops. Then again, I will save it as combine glucose, and I will save it as CSV. Okay. Okay. So now we have built the model for KNN. Well, you can explore the other one. We can build it with SVM. Just a click of button, then you build. Then okay, this is SVM. So then again, now then it will show that this one, the prediction is everything 100%. Okay, now here's the thing, okay, is that the different algorithm have some bias. So even though when we always talk about machine learning or AI or whatsoever, right? So you think that it is unbiased, it is actually not unbiased. It depends on what we feed it. So okay, in this case here, why do you get all this accuracy, which is all 100%? Some of the algorithm, they would try to do whatever they can to fit it in. So, but that is what we call the overfitting. So sometimes it could be good, sometimes it could be real, sometimes it could be just an artifact. So anyway, so we will try to look at that later on. So anyway, so this is the data from SVM. So they have mashed a bunch of genes that is predicted that could, you know, classify the tumors based on different subtype. If you look at the expression of this number of genes, then you can actually predict it. Okay, so we will close this, and you can equally do another analysis with random tree. Again, the same. You just press the build button, and then it will build for you. Okay, this one take a little bit longer because it's more complicated, the random trees. Okay, that's done. So this is random tree again. Well, it predicted 100% basically, and then with even less number of genes that could be predicted. Okay, so now we have built three models. Okay, the question is that if we give them an unknown, I mean the expression profile of unknown samples, how do they classify into the different subtype? So then this is exactly what I'm going to do. I'm going to open another data set, the one that we just did with the combined data set, the CCLE and also the ICGC. All right, I'm going to open this again. The matrix here, and matrix is here. This one here. Right, so this is so this is the combined data set that we have. So you have 4 different class and also the CCLE, which is the cell lines. Okay, so now what I'm going to do is I'm going to ask the computer to classify for me, which cell lines belongs to what subtype. So I go to classify here. I'm just going to load the model that I built just now, and then click the apply button here. Then it's done. So, now how do you visualize it? So there is a predicted class. So now if you look at that, so you have 4 class now, including the CCLE data. So if you want to look at the panel that I generated, the one that I displayed was actually using heat map. Oops, sorry, heat map here. So now, so I'm going to do a little bit of the filtering as well. Let's see. So the sample, so the variable is the genes, and then the sample order is by class. Okay, so I'm going to do a hierarchical clustering over here. And the order of this is the predicted class. So now you can see that. So I'm going to overlap here. On top would be, I don't need the label at the bottom, whereas I need, on top variables, the samples. So, what I have here, this is the original class that we put it in. So the go to here, whereby the pink color is a subtype 1, and then the yellow is a subtype 2, and then 3, and then 4, and then the red is the CCLE cell lines. Okay, so the predicted class, now they give you a different color scheme, so that's why it doesn't match. So now if I change this color, one is subclass 1 is pink, right? Okay, then followed by, then number 3 is what? 3 is blue, and then subtype 4 is white. Okay, so now then here, then you can see is that while most of the subtype 1, okay, when we run the prediction, it predict quite well, and then for the subtype 2, well, there's a few, okay, subtype, okay, so the red is a CCLE here, okay, so, and then you have a bunch of a red here. Now, here you have a little of 2 samples here, which is which is classified as the subtype 3, but in our analysis, well, it is predicted to be subtype 4. So it's pretty close that way. So now, so that's good. Now, if I want to look at the samples, okay, G90. Okay, now that if I want to look at what cell lines it belongs to what, so you can see that, okay, these are all the ICGC tumors. If I blow it a bit bigger, you see down here, so these are all the ICGC tumors. So this one, the part 2, whatever, 89885, so which is here, it classified as the subtype 1. Okay, now for subtype 2, you have 3 cell lines here, SNU324, YAPC, and also this 3. And then for squamous, you have a bunch of cell lines. And then for the this is subtype 4, you have more cell lines here. But of course, this is based on the KNN analysis. So similarly, you can build the same, okay, using the model that we built for SVM. Then you just need to apply it. Okay, so now it gives you something what we call the, okay, so class 1, so class, so now you have, this is the original subtype that we based on the Nature paper. This one is based on KNN, and this class Likert one is based on SVM. I'm going to build another one, which is by the random tree, and then apply again. So now you have 3 different predicted. So I'm going to display this on this graph here to see how close that they predict each other, basically. So I'm in class 1, class 2, class 3. Okay? So, but the one problem with this is almost every time that, okay, yellow, pink, white, red. Oh, so, sorry, have to follow this pink, yellow, blue, white. Okay, pink, yellow, blue, white. So for the other one, it's also pink, yellow, blue, white. Okay, so now you can see how close they are. So most of them are predicted quite well. basically, right? So all the tree model. So now this one as well. Now you will see that some of the cell lines, okay, might appear different by different prediction. So that is expected. That's why you have that table there. Based on each prediction, what is the core that they come out, whether they are squamous or whether they are progenitor and so forth. Okay, so anyway, so this is how you can do the analysis to predict the cell lines, whether they belong to what cell type it is. Okay, so that's it pretty much.